morning. Thank you very much for being here. It's a big pleasure to introduce you my very good friend, David Patterson. David does not need to be presented because you all know him, but I will have to present him. So David, at this moment, is professor at the university in Singapore. I don't know the name of the university.
National University of Singapore.
Okay.
Yeah.
And also is director of the Advanced IDs, an organization working on developing trials and studies in clinical research, mostly in antimicrobial resistance area. David is a very well-known expert and very influential person in the treatment of gram-negatives, mostly. He's developed many good studies. He's been working in Australia. He was also for a while in Sicilia. He's been also working in Pittsburgh, came back to Australia, and now to Singapore. He's been very influential for all of us because he was an investigator of a study that you all know. It was the Merino trial. That trial was a landmark from trials in gram-negative treatment for antibiotic treatment for gram-negative bacteria. We all tried to follow his path. We developed the Forest trial and the Lancet trial after that, but that was really a landmark trial. So it's a big pleasure for us to have him here. He's been in Seville for a couple of days, and we hopefully will have him come back in the future sometime. So
Exactly. Muchas gracias, Jesus. And it's lovely to be here. I feel very ashamed that this is the first visit I've made to Andalusia, and it will not be the last one, for sure. So back in 2006, so 20 years ago, Jesus came to visit me in Pittsburgh, and then later, maybe 2014, I can't remember it precisely, also in Brisbane. And Pilar came to visit in Brisbane for 3 months or so. I know Emanuele and Belen I've been a fan of her work, but you should be aware that the work you do here in Sevilla, when you publish it on a Tuesday, in fact, the whole world changes what they are doing on the Wednesday. It is true that when you publish important research from here, it does have a global impact. So this is really an amazing team that you are all together. So my interest is in gram-negatives. And when I was in Pittsburgh, I was doing more molecular epidemiology, risk factors. But one area that I was not really aware of was the ability for clinicians to do clinical trials. And that's really my passion now. And I think it is the area where we can really change clinical practice the most efficiently. I do some work with pharmaceutical companies, which I do want to declare. Like I mentioned, I am very interested in clinical trials, and every 2 weeks, my group puts out a newsletter on the most recently published clinical trials, and we now publish it also in Spanish. So if you are interested in getting this newsletter every 2 weeks, feel free to scan this QR code, and then you can get it, and you can elect either to have the English or Spanish or even the Chinese version. So maybe you prefer the Chinese version. All right. So as Jesus mentioned, I'm now in Singapore, and Singapore is a small country, and in fact a small city. It's quite wealthy. I think there are more billionaires in Singapore than any other city except New York and London. So it's high income, high standards of healthcare. There is a seat in the front, and I promise I won't ask you a tricky question. But the reason I came to Singapore was that I wanted to do clinical trials where antibiotic resistance in gram-negatives was most prolific. And so we now have a clinical trial network with more than 100 hospitals across Asia. And I just wanted to give you a little bit of a flavor of what infectious diseases in Asia is like. Well, first of all, it's important to realize that the populations in Asia are huge. So last year, I was invited to give a talk in a small Chinese city called Hefei. I had never heard of Hefei. They said, oh, it's just a small regional city, 15 million people. And, the hospitals have over 3,000 beds. It is really amazing, the populations in China and India, Indonesia, etc. So some of the healthcare in Asia is like you would find here, very modern. And I wanted to just sort of start by giving you some experiences, for example, in China, because we now have been able to start clinical trials in China. I told Pilar a sad story from China. The first clinical trial we started there, and I feel a bit embarrassed telling it in front of people. The first day the hospital was activated to do a clinical trial, in the first hour, there were 3 randomizations. And I thought, this is fantastic. We're going to recruit so many patients. But then I looked closely and they were the same age and gender, the same source of infection. And they randomized once and they didn't like the therapy. They randomized again, they still didn't like it. They randomized a third time and, this is the one we want. So that afternoon, one of my colleagues got on a plane and went there and, okay, we're going to start again. What does it mean to do a clinical trial? So yes, the Chinese have not had a lot of experience, certainly with investigator-initiated clinical trials. Now, you probably know that universities around the world put in rankings of which is the best university in the world. The university here, do they concentrate on these?
300.
300, okay. So, you would expect Oxford, MIT, Harvard, Princeton, Cambridge to be high. But you maybe have not heard of Tsinghua University and Peking University. And you can see they're number 12 and 13 in the world. And it's quite strange because they are both in Beijing and there's a fence between them. So they're basically on the same very large, well, it's actually 2 campuses, but if I was, if I'm a Betis fan and I'm a Sevilla fan and there's just a fence between, I'd probably shoot a drone or something. But they seem to have a healthy competition. But I expect we'll see more and more of the Chinese universities really come very strongly in these world rankings. Where I am at the moment, National University of Singapore, number 17. So we're quite, it's a well-resourced university. It was interesting, ARLG from the US did a comparative evaluation of outcomes of Acinetobacter infections in different parts of the world. And 30-day mortality from serious carbapenem-resistant Acinetobacter infections in the US was 25%, and in China was 20%. So they do, in the big hospitals, have a very high standard of care. And it's something that we should be aware of. It's tricky because we see all of these Chinese names and it's hard for us to differentiate, are they from a good place or a bad place? But the good places do great clinical work and have a lot of capacity for good research. India is quite different compared to China. Obviously, it is another huge country, over 1 billion people. Again, they have some very modern, amazing hospitals. But then there's situations, even in New Delhi, they do not have an automated blood culture detection. They do a blind subculture every 3rd day of incubation. You know, in New Delhi, I was just shocked that sometimes the standards are not good. Looking elsewhere in Asia, you know, countries like Malaysia and Indonesia and Thailand, there certainly are places of wealth and high standards. But for example, if you have an outbreak of dengue, I've also seen this with leptospirosis, suddenly you have a hospital that has 2,400 beds, but you have 3,600 patients. So patients share a bed or they sleep on a mattress between other beds. I've seen situations in the intensive care unit where relatives sleep under the bed of the ICU patient. It's really very different from what we expect. Now, this is the saddest thing I've ever seen in my trips in hospitals in Asia. This is called a rabies cell. So I'm sure if we ever had a patient with rabies, we would sedate them, ventilate them. They'd be in a quiet room in the ICU with comfort care. In low-income settings in Asia, if a child, and normally it is a child, has rabies, they go in the inner cell and the parents stay in this outer cell. There's no sedation. The parents pass them food as required. And unfortunately, no child who goes into the rabies cell comes out alive. So it really is a very tragic thing, particularly in such a preventable infection. On the bright side, almost every hospital I've been to in Asia has a hospital cat. And so this is from the male surgical ward in the Philippines General Hospital. And one of my colleagues said, "Oh, isn't it bad to have a cat? Wouldn't there be Bartonella, Pasteurella, what other things? Toxoplasma." But I guess they eat the mice and the rats and the patients can pat them. So maybe it's not such a... Actually, Emanuele, there was a hospital when I worked in Sicily, I remember going to a hospital and there was a dog that just would walk through. It seemed to own the hospital. It did have scabies and... But anyway, maybe I don't think in Andalusia. No, that's good. All right. So in my explorations in Asia, there are 3 interesting things that I've observed with gram-negatives. And we know now with infectious diseases that something that happens in a city in China or maybe in India, it happens on, March the 16th. By March the 21st, it's possible it could be here in Sevilla or elsewhere in the world. So I wanted to talk about 3 emerging threats in antimicrobial resistance in Asia in gram-negatives, and also a little bit about what are some new treatment options related to these. So the first one is hypervirulence in Klebsiella and its convergence with multi-drug resistance. So in Asia, and I'd be curious to know if you've ever seen this here, this syndrome with Klebsiella pneumoniae, where they have liver abscess, they have a very hypermucoid, sticky Kleb, Have you ever seen that here? Yeah. And was it in someone who'd been from Asia or a local person? Yeah. No, it's interesting that this is a person with endophthalmitis from Australia. You know, there are certainly these cases that we're now seeing in other parts of the world. We know that hypervirulence in Klebsiella is a real entity. It does have adverse clinical outcomes. And last year, the WHO put out a special disease outbreak alert describing the confluence of hypervirulence in Klebsiella and multi-drug resistance, including carbapenem resistance. So my clinical experience, though, has been these carbapenem resistant Klebs with these hypervirulent genes don't present with liver abscess and endophthalmitis. They seem to have a worse ICU outcome. Now, whether it's because they're already in the ICU or whether it's the virulence of the organism, we haven't quite determined. It's interesting, it's been found in the US. There's been descriptions in people coming from the Ukraine war. And so, you know, it is something to be alert to as, you know, is it actually emerging outside of Asia? You know, why, for example, did the patients here in Sevilla or in Brisbane, who we don't believe had any travel to Asia, why did they acquire these hypervirulent Klebsiellas? And is there something different about treatment? So, you know, for example, with group A streptococcus, if you have hypervirulent group A strep, you know, we may add clindamycin, for example, to turn off toxin production. Is there something different or additional we should do with hypervirulent Klebsiella in terms of treatment? So I'm sure some of you have been in this situation, maybe Jesus and Pilar, Ellen and others. Someone invites you to write an article, and I thought, oh, this is not going to take very long. It was antibacterial agents in phase 1, 2, and 3 clinical trials against gram negatives. And I thought, oh, there'd be 3 or 4. It'd take me an afternoon to write it. But I found out there's actually 47. And it took, I went to Bali with my family, and I ended up spending a week in Bali writing this paper instead of getting bitten by monkeys or but I found there is a you know, with phage therapy, phages have lysins that I think disrupt the cell membranes, and this group from India has actually synthetically engineered the lysins of phages that are active against Klebsiella. And so they are about to start phase 1 clinical trials of a Klebsiella-specific therapy. So it would be interesting, and it's interesting to think about how you would do a clinical trial, because I presume we would want to continue standard of care therapy. You know, if there's a KPC producer, you'd probably want to keep going with ceftazidime avibactam, but you'd probably have to do a superiority trial, say ceftazidime avibactam plus the clebacin versus ceftazidime avibactam alone. And presumably you'd have to need a rapid diagnostic, so you wouldn't want to wait, you know, 3 days to have, not that it takes us 3 days anymore for identification, but it's interesting just to think about how you would do a clinical trial to try and show that there's something specific against Klebsiella that may provide some assistance. So the second emerging issue I wanted to talk about was changes in penicillin binding proteins in E. coli. And before you fall asleep, because what are you talking about? Maybe I'll even make you fall asleep even more by, let's just sort of think about what are the mechanisms of resistance to beta-lactam antibiotics. So in gram negatives, you know, we think everything is beta-lactamases. And actually, Jesus, are there any students here who I could torture briefly? Or maybe anyone can answer, you know, what is the IQ of a bacteria? What is the IQ of a bacteria? So, you know, of course, bacteria don't have brains, they don't have an IQ, but it's amazing that in response to the threat of beta-lactams, bacteria have developed this resistance mechanism, the production of enzymes, beta-lactamases, that can destroy the beta-lactam ring. And so, you know, we have really, and even today, we always think of beta-lactamases as the main source of resistance. Sometimes there might be loss of porins or outer membrane proteins, and think of, for example, OPRD, you know, Pseudomonas and imipenem resistance. Sometimes there are upregulated efflux pumps that contribute to beta-lactam resistance. But we never really think of changes in penicillin binding proteins because, of course, if I'm a beta-lactam antibiotic, I've got to get through the outer membrane through a porin channel. I've got to get through the periplasmic space where there's the beta-lactamases, and I've got to bind to the penicillin binding proteins, which are on the inner membrane. Now, of course, with Staph aureus, with Enterococcus faecium, for example, you know, we do think about changes in penicillin binding proteins. But what has been noticed is that particularly in PBP3, there are changes that change the ability of antibiotics that bind to PBP3 to work. And it's interesting, antibiotics like aztreonam, ceftazidime, cefepime, cefiderocol preferentially bind to PBP3. Carbapenems bind to PBP2. And what has been shown is that there is frequently now, unfortunately, in NDM-producing strains of E. coli, an insertion of 4 amino acids into PBP3 that reduces the affinity of beta-lactam antibiotics to PBP3. And as I mentioned, unfortunately, it's also in strains that are producing beta-lactamases. So our friends, Patrice Nordman, Laurent Perel, have looked at the effects of this. And if you have ceftazidime, aztreonam, cefepime, those MICs are higher. Also, if you are combining them with a beta-lactamase inhibitor and also cefiderocol. So this does have a potential implication on our therapy. In India now, probably 40% of E. coli have got this PBP3 change. So it is really taking off big time there. We're seeing it now throughout Asia. It would be interesting to evaluate whether in Andalusian E. coli you're ever seeing these changes in PBP3. And unfortunately, I guess they're going to be in ESBL producers, AMP-C producers, or carbapenemase producers, particularly NDM. So again, just luckily in my search, I found that there were some antibiotics that bind preferentially to PBP2 in development. And the advantage of this is if there's these PBP3 changes, we don't care because we're binding to PBP2. And so one of them is called cefepime with zidibactam. And so zidibactam is interesting because it is a beta-lactamase inhibitor, but it is also able itself to have antibacterial activity by binding to PBP2. So it has actually gone for FDA approval. There's another one called nacubactam where they're going for European approval. So these do have excellent activity against MBL producers like NDM, for example. So it may be that there are new options if we do have these E. coli isolates with these PBP3 changes. But as I mentioned, in Asia, this is taking off big time. And so, you know, even now when we think about, so last week when I was in Italy, in every hospital I went to, they've started to use aztreonam avibactam for their NDM producers. If it was an E. coli PBP3 change, aztreonam avibactam is less effective. Finally, I want to talk to you about carbapenem-resistant Acinetobacter. Maybe 10 years ago here you would be interested, maybe today you're not. Is it still the case that, that is amazing, yeah.
11 cases in the whole week.
Wow. So you tell me if I'm wrong
9
on,
million inhabitants.
that is brilliant, yeah. But did I tell you I didn't wash my hands this morning? I can, if you're interested in carbapenem-resistant Acinetobacter, I can reintroduce it. Sorry. But my opinion is that it is highly susceptible to well-organized hospital cleaning. And 2 weeks ago, I was in northern Thailand, very close to the border with Myanmar. In a room this size, there would have been probably 16 patients being mechanically ventilated. There was one patient who was on continuous renal replacement therapy, and they brought in the machine, and I'm sure I could see mold growing on the machine. I am sure that if I touched any bed rail, any piece of equipment in that ICU, I would have got carbapenem-resistant Acinetobacter. I am sure that part of the issues with this is that hospital cleaning is neglected, unfortunately. And even in Australia, it'd be interesting to know what happens in Andalusia. The lowest paid workers in the hospital are the hospital cleaners. And it wouldn't be a problem here, but often they are from Colombia, Philippines, you know, other countries where their English is not great. They're just trying to get a job to survive in Australia. And unless they are given clear instructions they can understand, they don't clean. So we did introduce a thing where we put the, do you know, like the glow in the dark thing on different surfaces in the hospital to evaluate the compliance with cleaning. And so they'd cleaned, and then we could see whether, you know, what percentage of efficacy. I do have a question. Even though you came late and I said I wouldn't ask you a question. In this evaluation, what is the cleanest place in a patient's room? The place where cleaners always clean it beautifully. So
I
in,
think is the table close to the patient.
no, the toilet seat. You know, every cleaner knows.
I'm not agreeing with that.
Yeah, but certainly in Australia, every cleaner knows they've got to clean the toilet. But the bedside table or the bell to ring the nurse, not cleaned. We did have one clever cleaner who, you know, to show these spots where you've cleaned, you have to have a little UV light. And one of the cleaners himself bought a UV light detector. So he just cleaned where we put the spot and nowhere else. So unfortunately, outside of Underluthia, carbapenem-resistant Acinetobacter is still big and important. And, you know, when you look at the IDSA guidance, they talk about sulbactam as being a principal part. And part of the reason they talk about sulbactam is even though we think of it as a beta-lactamase inhibitor, it actually binds really nicely to the penicillin binding proteins of Acinetobacter. And the trouble, though, is that sulbactam can be destroyed by carbapenemases and also other beta-lactamases because it is actually a beta-lactam, even though it is also a beta-lactamase inhibitor. And so I don't think sulbactam durlobactam is available in Spain, but when I was in Milan, they had some of the teenagers who'd been in the fire in Switzerland on New Year's Eve, and they imported sulbactam durlobactam because these were burns patients. They had Acinetobacter infections. So if, and I hope you don't, but if you have serious infections with Acinetobacter, in my opinion, sulbactam durlobactam is the treatment of choice if you have it available. So they compared it with colistin. There was an improvement in mortality. It wasn't statistically significant. Their clinical improvement, microbiological outcomes at the end of therapy were statistically significant in favor of sulbactam durlobactam, and it was also significantly less nephrotoxic than colistin-based therapies. The trouble is that around the world, it is only available commercially in the United States and China. It's expensive. It is recommended to be used with a carbapenem. And there are now probably 5 to 10% of Acinetobacters which are producing NDM. And the durlobactam doesn't inhibit NDM because that's a metalloenzyme. And again, this story about mutations in PBPs can be an issue. So what is new in Acinetobacter treatment? So remember that in the AIDS time, we used to use oral rifabutin. So some people have made rifabutin into an intravenous formulation, and it gets very high concentrations in the blood and the lung and is able to inhibit Acinetobacter. It's also synergistic with polymyxin B or cefiderocol, and so it is being trialled as a combination therapy. And that's starting phase 3 trials very soon. Roche has a peptide that is specific against Acinetobacter called zosurobalpin, and it's scheduled to start phase 3 trials next year. So by the time Acinetobacter comes back to Underluthia, you may have sulbactam durlobactam, you may have at least 2 other options. All right. So if I was to give you my opinions today about treatment of MDROs. Now, feel free to send some poison darts my way. First of all, for ESBL producers, and I'm going to be specific here, for example, in the blood. The purpose of the Merino trial was to show that if we had started someone on piperacillin tazobactam and it turned out to be an ESBL producer and it was susceptible, that we could continue piperacillin tazobactam. So of course, we were disappointed because our hypothesis according to our data was wrong. Now, at ESCMID this year, Mikhail Paul is going to present some of the results from the Peter Penn trial. So that will be really interesting. I haven't seen them yet. My expectation is that piperacillin tazobactam will be much closer to meropenem, but let's wait and see. If we have KPC producers, I think ceftazidime avibactam or meropenem vaborbactam. Some people say meropenem vaborbactam gets better pulmonary penetration. OXA48, ceftazidime avibactam. NDM producers, our game changer trial results were disappointing with MBL producers. And I think aztreonam avibactam would be the preferred therapy. Have you had to use it here yet?
And the drug that we have not used.
Right, but it's
Which one?
aztreonam avibactam.
Well, we are using.
Okay.
I come from another city.
Ah.
Very close to it.
Yes.
And we are using a aztreonam.
Yes.
The cases with the VIN, with the DNA.
We had an outbreak of in one producer that was resistant to all the drug. It was avibactam plus aztreonam.
No,
So I do have some experience with Vim producers. So in the old days, and this is advice to you who like to present things at ESCMID, for example. So when I was in Palermo, we had an outbreak of Vim producers, and I sent an abstract to ICAC saying successful control of outbreak of Vim producing, it was actually Pseudomonas, and it was accepted. And basically the day after my abstract was accepted, the infections, the Vim producing infections came back. And I thought, oh, will I withdraw the abstract? But I went ahead and presented it and showed the data up to the date of the ICAC abstract submission where it had gone away. And the next slide was it's come back. So, you know, we have to always be prepared with our ESCMID abstracts to, you know, what you present in the abstract may not be the final data. I'm very curious to hear your perspectives on carbapenem-resistant Pseudomonas. There's not been a head-to-head trial of ceftolozane tazobactam and ceftazidime avibactam, imipenem relibactam, cefiderocol. You may be aware of the TREAT GNB platform trial that we're going to be doing together, because here is an opportunity to evaluate these drugs as potential comparators. But my tendency would be to use ceftolozane tazobactam, but there's not clinical trial data. And finally, carbapenem-resistant Acinetobacter. If I didn't have sulbactam, durlobactam available, I would be using cefiderocol, but in combination with something else. Now, is this going to change in the future? So we have to wait for the publication of the Astarte trial. So maybe we'll have temocillin there. Maybe we'll have some other carbapenem-sparing options. I think with NDM producers, it's likely that this issue with PBP changes in E. coli will become more pronounced, and we'll have some other options. They are evaluating cefiderocol with an NDM inhibitor called zerubbabactam, so maybe that will become available. And finally, with carbapenem-resistant Acinetobacter, there are some new options coming into clinical trials. So I think for those of us interested in multi-resistant gram-negatives, the problem is not going away. The problem is evolving. The bacteria are keeping on acquiring resistance mechanisms and creating new troubles for us. But I think, you know, it has been an interesting time to do clinical trials over the last 10 or 15 years. But the next 10 or 15 years, thinking about future strategies for treatment of resistant gram-negatives and how we can optimize treatment, but at the same time prevent ecologic damage, is certainly going to be an interesting opportunity. So thank you, Jesus, and thank you all of you for coming. I'm sorry I couldn't deliver this in Spanish, but hopefully you could make sense of how I was thinking. So muchas gracias.
Time for some questions. So David, I have a good opportunity to have him.
Yes.
First is a comment. The insertion in the PDP3
Yep.
mutation in E. coli, when we had cephalosporin available for testing, for microdeletion testing, we observed that we had already before the use of cephalosporin in strains from Macarena or from the region, they were before the use of cephalosporin. I think there is a polymorphism, but there is a chance to have E. coli mutation. So I don't think it's something that emerged only because of the call. I think they were before. But I cannot have the data or figures of that.
Yeah.
And regarding OXA48, most of OXA48 that we have very low MIC for meropenem. Very low. I mean, it could be 0.5 or even less than 1, most of them. So we can use meropenem buffer for that because our fans are very strong inhibitors of the SBL. So in those cases, what do you think about? In those cases that we have a CTX1015 or other ESBL plus OXA48, and we can inhibit it with bavoractan, the SBL that can increase a little bit the MIC and use meropenem for the OXA48. So this combination is also available for OXA48. I mean, not only reduced for PPC.
Yeah. So just for the people remotely, the question was about treatment of OXA48, given that frequently the meropenem MICs are very low. Could I ask then, why would you add Vaborbactam? What is it really? Because the meropenem is stable to ESBLs.
When you use Vaborbactam, you can reduce a little bit one of dilution or two.
Okay.
Because the ESBL
is being inhibited as well.
That's my
it would be interesting to put it into the TreatGNB algorithm.
We have meropenem only, meropenem alone for those specific cases, but we didn't thought about meropenem.
I tried in a synergy with, and they have a very, very, I mean, meropenem Vaborbactam in OXA48 has very, very low MIC.
Yes.
Sometimes similar to meropenem and sometimes lower.
Yes. And just to reflect on your initial comment about cefiderocol already having a distribution of MICs. So again, I need to apologize. So when I first saw the MIC data from Shionogi, it looked fantastic, so fantastic for gram-negatives. And they also had the trial data from their complicated UTI trial where it was superior to imipenem. So I went back to my hotel room, again, this was ICAC, and I tried to write down, because we talked to them about doing a trial. And Emanuele, you know, the most important thing about doing a clinical trial is having the name. And so I was trying to think, what is a word that means something that's going to be a big step forward? And I came up with game changer. And of course, the results were hardly game changing. So at least with Astarte, you know, whatever the results, it's not going to be a bad reflection on Astarte. But one of the issues with cefiderocol, of course, is the susceptibility testing, that you have to have the iron depleted media. But I think your comment is really, it's not just cefiderocol that has been binding to PBP3, you know, cefepime, ceftazidime. So we've had 20 or 30 years of gram negatives getting their PBPs getting exposed to antibiotics. And it's not surprising that over that period of time, they have been able to mutate so that they can resist these antibiotics. But it is interesting that cefiderocol is by far, it's not a perfect antibiotic in vitro at all.
Yeah, David.
Oh.
Thank you very much for your excellent talk. It's incredible to have you here today. I would like to ask you a few questions. First, what advice would you give to your younger self at the beginning of your career? And second, what do you value most in the people you worked and collaborate with?
Fantastic. Really great questions. So I remember calling my father when we discovered TEM 131. And who cares about TEM 131? Nobody. I convinced my dad that this was, got an AAC paper. So I guess when I started my research, like discovering new variations of beta lactamases, to me seemed that was research, but in fact it had zero impact on patients. And I think for those of you who are clinicians, and I would include clinical microbiologists as well, I think the research we should be doing is something that is going to change how we treat patients. Optimally randomized trials. If we can't do randomized trials, how can we design observational data so that it does, without too many confounders and bias, give us something that has a clinical impact? So my advice, if I had started research again, because I'm a clinician, I would have not done this, beta lactamase stuff, and I would have gone straight into more clinically oriented research. So that's the first thing. I had to apologize to Jesus this morning because I was late. And the reason I was late is there was an email from a colleague who was asked by another colleague to do something, and she didn't want to do it. I think the most important thing, looking at the group here today, is open communication and not hiding things, but being clear to everybody. A lot of what we call politics in research or universities or hospitals is because one person knows something, other people don't. There's this loss of trust. And I think if we communicate well with each other, I think that's the best way to be a colleague. And it's the same when we look after patients as well. If I've made mistakes and bad mistakes in medicine, and I think our patients and their families appreciate it when we are honest and say, okay, what I did was not the right way. and I apologize, rather than covering it up and eventually, it's worse when someone eventually finds out something and then they're angry. So I think to be a good colleague is to be open in communication is my feeling. But lovely questions, Belen.
My name is Julian Torres Cisnero. I'm coming from another city, and I'm glad to be.
Yeah.
Well, and of course, I'm be very happy to have you here today and hear your excellent talk. So thank you very much. And my question is regarding is almost a comment, and I would like to know your opinion about it. Is I think then we have paid a high cost to the overuse of carbapenems to cover ESBL producers in medicine. And the cause is the emergence of carbapenem resistant gram-negative bacteria. So I know that the carbapenems are very cheap, but I'm thinking about if it's time to change our policy and to use another options for these bacteria in order to reduce the burden of carbapenem resistance in our hospitals. And I don't think that the solution is to use carbapenems and carbapenems and carbapenems in many hospitals, in many patients, severe patients, critical patients. And for example, carbapenem can be indicated for a resident in an emergency room, and that's something that we have to change.
Yeah.
So it is time to have a policy in which can indicate another options than we are reserving now for multi-drug resistance and bacterial to route the antibiotics, to route the carbapenems, at least to reduce the use of carbapenems in 50% of patients. I think that we have to think about it.
Yeah.
And I would like to know your opinion. For example, I don't see other options in your talk like cefepime and methotrexate. We are introducing this antibiotic now in Spain with the indication of ESBL producer plus OXA48. And what about timipenem and relebactam in your options? And what about another option that we have in commercially available, at least here in Spain?
Yeah. So I think this is an excellent point. And maybe, when Pilar or Manuele came to the intensive care unit at Royal Brisbane Hospital, where I worked for more than 15 years, often when we came into the ICU, patients were on either piperacillin tazobactam or meropenem. That's it. There was almost complete homogeneity of antibiotic choice. So they would only have two options. And I think your suggestion of heterogeneity of antibiotic choice is a fantastic one. I don't know about cycling. In my mind, and I think there are some mathematical models that suggest that if you do have a wide array of antibiotics, heterogeneity of choice, that is what reduces emergence of resistance. So, as you say, you might have someone on cefepime and tazobactam, there might be someone on temacillin, there might be someone on ceftazidime and fosfomycin, there might be someone on meropenem. But instead of a high-risk unit only having two antibiotics going in the whole population to have a broad choice, I think is the optimal way. To me, what informs that choice should be randomized trials. And I've become more and more mainly through surprises. So, Jesus, I think you've been doing the SNAP trial with Staph aureus bacteremia. My whole life, I've been using flucloxacillin for serious staph infections. And the SNAP trial has suggested that cefazolin, for a variety of reasons, is a better option. And I wouldn't have predicted that in a clinical trial. Maybe others would have. But I think if we are able to collectively do clinical trials that can inform our choice and provide, even a bigger array of choices than this. And, there are going to be some times when we use ceftriaxone or ampicillin, even if it's a gram negative. So I think we're agreeing, even though, maybe in retrospect, we were a bit heavy in our interpretation of the Merino trial and really pushed carbapenems as the choice. I think as we get more clinical trial data back, we'll be, but again, we shouldn't, just say ceftazidime and fosfomycin is a beautiful choice. We shouldn't be using that in every patient. We should have a variety of antibiotic choice. So I think the cycling, I maybe 15 years ago, people were looking at cycling of antibiotics, and I'm not sure it really came.
The problem of cycling here, at least
Yeah.
here in Spain, is that the alternative carbapenem are expensive.
Right. Yes.
And we are working in a public system.
Yes.
So to explain the authorities that is something good for the health, to spend money cycling carbapenems is urgent, in my opinion.
Right.
Something that we have to do.
Well, you're not talking about cycling, but mixing.
Of course, mixing.
Yeah.
Mixing, that's the word.
Yeah.
Mixing is not the after Merino trial. The overuse of carbapenems in all hospitals is amazing.
Yeah.
It's amazing. And at the same time, we are suffering the emergence of carbapenem resistant bacteria. So how combine these two things?
Yes.
My experience, we have a very high problem with the carbapenem, with KPC producing Klebsiella pneumoniae in my hospital.
Right.
Now it's controlled, but we have 5 years with a very high rate of this infection. How to control this problem if we don't control the use of carbapenem?
Yes.
In order,
Yeah. No, I think this is a good point.
Thank you, David, for a very nice presentation. Regarding the question made by Belen, I would like to make a comment and to know your opinion. I think one of the factors or important factors in the success of the clinical trials developed by Jesus and his teams is that behind every clinical trial, there was a combination of clinical microbiologists and ID people designing and developing the clinical trials in such a way that behind the clinical trial, there was a powerful microbiology. I would like to know if this is the same experience with you and what is your opinion?
Yeah. So exactly the same. And so Patrick Harris and myself both trained in clinical microbiology and worked in clinical microbiology as well. And the need to, you know, we have this amazing opportunity. We're not just we're randomizing patients, but each patient has this little pot of gold, which is their bacterial isolate, which can help explain our clinical trial results as well as get further insights. And there may be, you know, increasing aspects like rapid diagnostics that may, you know, in the Merino trial, we were criticized for waiting 72 hours. You know, you could randomize up to 72 hours after the collection of samples. If we want to incorporate rapid diagnostics into our clinical trials, we have to have the involvement of the clinical microbiologists and the clinical microbiology laboratory. We also need to consider, I think, pharmacists, pharmacologists, PK/PD people, because you know, now we're really understanding the importance of infusion time and dosage and frequency in our therapies as well. And what in Australia is an important thing, and I'm not sure in Spain, is we are now asked to involve the community or patients in the design of our clinical trial. Has that come forward in Europe as well? Yeah. So it is not just, and the other thing is, I used to think clinical trials was all about the idea. But in fact, now I realize that 95% of it is the clinical trial operations. You know, how is the trial, you know, you've got to get agreements, you've got to get budgets, you've got to get, you know, monitoring, you've got to manage your data. All of that is not science, and yet I think that's probably 95% of clinical trials, and something that we sort of, you know, I used to think the only interesting thing about clinical trials was designing it and then getting the results, and all the stuff in between was boring. But in fact, that is just so crucial. Otherwise, your trial's not going to recruit well. Yeah.
An important part of the team is dedicated to managing the
Fantastic.
trial.
Yes.
How important.
Yeah.
So one question. So I think that the Game Changer trial, to me, is a very interesting trial. So again,
it's not perfect, and in retrospect, we would have changed things. But yeah,
it's not a perfect trial as far as I know. No, but I mean, it's the results for cefiderocol are disappointing. But I think there are two aspects that are quite relevant. One is that cefiderocol is not that bad for Acinetobacter, as a credible trial seems to show.
Yes.
And cefiderocol is not such a good drug for NDMs as it was supposed to be. So both aspects are interesting. So I would like to know, what is your thought about it?
No, exactly. And subsequently, well, actually, during the course of the trial, Robert Bonomo and others have shown that there are circumstances where NDM can inhibit cefiderocol. And so it does make sense that it's not necessarily such a beautiful drug against MBL producers. Acinetobacter, you know, the numbers weren't great. I think there's still uncertainty. I have to say I would be nervous using cefiderocol by itself for Acinetobacter, but I do have more security now. You know, it's not a dreadful Acinetobacter drug. But yeah, it's for sure the last time Shionogi will ask us to do, you know, run a trial because they did not like the result. Maybe, I hope there's no Shionogi people here. When I gave a talk similar to this in Italy last week, some of the Shionogi people were there, and they, not happy. But I think that's the importance of investigators doing clinical trials, because if they're just the regulatory trials, we don't get the information that we really want for our clinical practice. Yeah.
And even when trials do not provide the hypothetical result that you thought, that they are so interesting to analyze.
Exactly.
We learn so much from them, from going into detail. From them, that I think that is really well. Okay.
So, David, thank you very much
My pleasure.
for being here.
Okay. Really lovely to talk to you.
Nice
Yeah,
to be
yeah,
here.
yeah.
So thank you very much for
Yeah.
coming.
No.
I know that we have